Bismillahirrahmanirrahim, and welcome back to this next video. And this is another video in the series of videos on the isotypes of the antibodies. In my previous video, I've told you that the isotypes or the different classes of the antibodies, they are based on differences in their heavy chains. And we started our discussion on the first type of the immunoglobulin that was known as the immunoglobulin G, and then we were talking about the different subclasses of the immunoglobulin G. If I give you a summary of that, I've told you that the immunoglobulin G is the most abundant antibody in the bloodstream, accounting for about 75 to 80% of all circulating immunoglobulins. And there are four distinct subclasses of the immunoglobulin G. They are known as the immunoglobulin G1, the immunoglobulin G2, the immunoglobulin G3, and the immunoglobulin G4. And these subclasses of the immunoglobulin G, they were based on differences in the constant region of the heavy chains. And whenever we talk about the immunoglobulin G, we are talking about the gamma heavy chain, because this is the type of the heavy chain that defines the immunoglobulin G. And these are differences in the constant region of the heavy chains that is defined by different things. In different subclasses of the immunoglobulin G, you are going to see differences in the amino acid sequence of the constant region. You are going to see differences in the length of the hinge region. You are going to see changes in the amount and the type of glycosylation and their biological properties. We discussed that in our previous video. Now, in this particular video, I want to focus on the first class of the, or I would say the first subclass of the immunoglobulin G, which is known as the immunoglobulin G1. Now, this immunoglobulin G1 is the most abundant subclass, accounting for about 60 to 70% of all immunoglobulin G in the human serum. So, among the different isotypes of the antibodies, immunoglobulin G is the most abundant. When you talk about the subclasses of the immunoglobulin G, the most abundant subclass is the immunoglobulin G1. Now, this is the general structure of the immunoglobulin G1. And if you look at the heavy chain, now this one is your heavy chain. This particular heavy chain is known as the gamma 1 heavy chain. So the gamma chain is defining that you're talking about the immunoglobulin G. Now this one is defining that you are talking about the first subclass of the immunoglobulin G, that is the immunoglobulin G1. So the heavy chain in immunoglobulin G1, that is known as the gamma 1 heavy chain. Now, as I've told you that one of the major differences between the different subclasses of the immunoglobulin G, that is the length of the hinge region. So when you look at the length of the hinge region in the immunoglobulin G1, that is 15 amino acid long. Now these black strips over here, they are actually representing the hinge region, which actually connect the FAB to the FC region of the antibody. Now these hinge region, they are responsible for connecting two heavy chains of an antibody. So they usually do this by the formation of the disulfide bond. So when you look at the immunoglobulin G1, there are two cysteine in the hinge regions, and they are responsible for making a single disulfide bond. So in the immunoglobulin G1, the two heavy chains, they are connected to each other by a single disulfide bond. These are one of the important properties that you are going to see in the immunoglobulin G1, that they are the most abundant one, and the hinge region length is 15 amino acid long. Another important property of the immunoglobulin G1, that is the glycosylation. Glycosylation simply means the addition of a sugar molecule. When we talk about the proteins, after they are made, they go to a process which is known as the post-translational modification. And in post-translational modification, you are adding a different kind of the molecules to the proteins. When we talk about the glycosylation in the process of the post-translational modification, that would be the addition of a sugar molecule to the protein. In this particular case, the protein is immunoglobulin G1. So what you do is that when you are going for the glycosylation of the immunoglobulin G1, you are adding a biantennary complex glycan to it. Now, this biantennary complex glycan, this is the journal diagram representing the structure of the biantennary complex glycan. Why we call this is the biantennary? Because if you look at in the structure, this is one antenna-like structure. This is another antenna-like structure. So this is for the antennary. Is they are two. Therefore, we call this is the biantennary. If you look at the composition of this biantennary complex glycan, you are going to see a lot of sugar molecules. For example, if you talk about these blue boxes over here, they are actually the N-acetyl glucosamine, and you are seeing a lot of them in the biantennary complex glycan. Then, if you talk about these green balls over here, they are the mannose. If you talk about these yellow balls over here, they are actually the galactose part of the biantennary complex glycan. Then you have got, if you look at these purple one, which are present at the end of the biantennary complex, they are the N-acetyl neuraminic acid, and they are actually present at the end of the biantennary complex glycan. Then at this particular end, you have got these fucose sugar molecule over here. So this is the biantennary complex glycan structure. Now, if you look at these particular terminologies over here, so the fucosylation, that would be the addition of a fucose. The galactosylation would be the addition of the galactose. And the sialylation, that would be the addition of the new 5-EC. Now, where does this biantennary complex bind in the immunoglobulin G1 molecule? So, if you look at it here, they usually bind to the constant region, the second constant region of the heavy chain of the immunoglobulin G1. So, you can expect the binding at this particular position over here. We will continue our discussion on the immunoglobulin G1, and in the next video we will talk about the different immunological functions that the immunoglobulin G1 can play.